Okay, so neurotransmitters in a chemical synapse are released into the synapse, but we don't want the neurotransmitters to stay in the synapse. So there's a couple of ways that the neurotransmitters can be either actively removed or they just kind of diffuse away. So the first one is probably the simplest, and that is that the neurotransmitters diffuse away from the synapse. And so there are characteristic neurotransmitters that have a certain form of termination that is more common. And so a good example of this are the endorphins. So endorphins have been in the news a lot lately because endorphins stand for endogenous morphine. And this is the drugs, the opiates, including oxycodone, which mimic the endorphins and bind to the same receptors. And so these are natural painkillers. So we actually can get endorphins through, for example, like exercise. So when you talk about people getting the runner's high, right, you can actually increase your endorphins. You can also get endorphins through eating spicy foods because the spiciness actually mimics the effect of pain and damage to the tissue. However, it doesn't actually cause damage. So it kind of tricks the body into thinking that it needs to release natural painkillers. And so spicy foods, including like, they create these spicy lozenges that people that have oral cancer or esophageal cancer sometimes suck on in order to reduce the pain that they feel from the sores that are in their mouth. And so that's kind of an interesting mechanism. But endorphins tend to diffuse away from the synapse, and they can have quite a long-term effect on the body. Okay? The second way that the neurotransmitters can be removed is by being broken down. So they can be broken down in the synapse. And so a good example of this is a neurotransmitter called acetylcholine. And we talked a little bit, or you may have learned a little bit about acetylcholine in lab, because acetylcholine is released between motor neurons and skeletal muscles. So this is the name of the neurotransmitter that is released between the motor neurons and the skeletal muscles. And so there is actually an enzyme that breaks it down. So acetylcholine is broken down by acetylcholine esterase. So this ACE ending here means that this is the enzyme that breaks down acetylcholine. So it is rapidly degraded and removed from the synapse because we don't want our skeletal muscles staying contracted. The other thing that acetylcholine does when we talk about the autonomic nervous system is that it actually slows down breathing and heart rate in the autonomic nervous system. And so we want to be able to speed up the heart if we need to. And so we don't want this neurotransmitter staying in the synapse for a long period of time. So this is kind of a rapid removal, a mechanism of a rapid removal. Now, there are some chemicals that we have actually developed as nerve agents that we use in warfare and also as insecticides. And so if we look at the nerve gas sarin, and the insecticide malthion. Right. Both of these inhibit the enzyme. So they would inhibit acetylcholine esterase from doing its work. And so it makes acetylcholine stay in the synapse. And so one of the things that the nerve gas does is it causes contraction of the skeletal muscles, but it also slows down the heart and eventually will just stop the heart and stop the breathing and cause death. So this is the nerve gas that they were storing out at the Umatilla Weapons Depot, but I think that they have successfully incinerated most of it. This is an insecticide that you would want to be very careful using. I had a student like 10 years ago tell me that he actually found out that the previous renters in his apartment had used this chemical all over their rugs in order to kill fleas. And it actually made his whole family really sick. And so you want to be very careful because this is, this is essentially a nerve agent. So when you use insecticides, you think, well, if it's going to kill the insect, my body works a lot like an insect does. Same neurotransmitters, right? Same essential organs. And so you want to be very careful when you use those. And the third way that a neurotransmitter can be removed from the synapse is it can be taken back up out of the synapse by the presynaptic neuron. So reuptake by the presynaptic neuron. And so a good example of this would be dopamine. Dopamine tends to be excitatory in the central nervous system. So we don't want too much dopamine. Really, there is homeostasis. So your neurotransmitters, if you think about it, it's kind of vicarious. You really want to make sure that your neurotransmitters are maintained in a homeostatic fashion. So anything that you do to disrupt the neurotransmitters can cause problems, specifically with withdrawals. And so a good example of this is cocaine, because this inhibits the reuptake of dopamine. So the high people feel from a cocaine rush is just all the action potentials that are being just generated in mass, right? And then what the body is going to sense is that dopamine levels are too high, right? And so the body is going to shut down dopamine production. And so you're going to have a withdrawal. So when you go off of the cocaine, you feel abnormal. So you take more cocaine to feel normal, and it takes more and more and more of the drug just to get you to where everybody else is. So that would be a good example of the reuptake with a illegal drug. But then we also have serotonin. So this tends to be inhibitory. And then we have what are called SRIs. So these are serotonin. This is just a squiggle. Reuptake inhibitors. So these tend to be antidepressants. Not all antidepressants are SRIs. But for example, some of the name brands that you might have heard would be like Prozac, Celexa, and Zoloft. So what that means is the serotonin stays in the synapse longer. And so it tends to reduce the number of action potentials, and it tends to make people who are anxious less anxious. So when we see somebody who's depressed, it looks like they're not doing anything. They're sitting on the couch. It looks like their nervous system is under-stimulated. But oftentimes what happens with depression is that they're sitting there and their mind is racing and they actually have too many action potentials. in the brain. And so if you increase the level of serotonin, that calms the mind so that they're actually able to get up and do something. So it's kind of weird when you think about depression and anxiety going hand in hand. It's kind of a weird connection. So again, actually, what's really kind of interesting about these is that you have to be really careful about going off of them, because if you go off of them, you can have side effects. And so your doctor actually has to help you wean yourself off of them. Otherwise, you're going to have some weird symptoms like maybe dizziness or nausea or anxiety attacks, that kind of stuff. Okay, so those SRIs do mess with the levels of neurotransmitters in the synapse as well. Are there any questions about those? So those are the three mechanisms of terminating the neurotransmitters in the synapse. So we're next going to talk about neurotransmitters, and the first thing I want to talk about is the categories based upon how they function. So we can talk about neurotransmitters that have a direct action. So direct action means that they bind directly to ion channels and cause them to open. To open. This tends to be fast. So this is faster. When we look at the indirect action, this is through a secondary messenger. So they bind to a receptor. Oops, sorry, I'm scribbly today. To a receptor that then initiates the response via a secondary messenger. And this is oftentimes what happens with cells, is that you have a receptor that then causes a chemical change inside the cell, and then that chemical change then leads to the response, which would be the opening of the ion channel. And so this tends to be slower in direct action. So let's look at the images in your textbook that describe these two mechanisms of action. Okay. So this would be where this is a chemically gated ion channel right here. And it's a protein that is floating in the plasma membrane. And then the green thing is the neurotransmitter, and it binds directly to it and it opens up the cell. Okay, so that would be a direct action. And then we have the indirect action, where it binds to a receptor. It's not the actual channel, but notice that it goes through all of these different chemical changes that eventually leads to a secondary messenger. And we're not going to talk too much about that mechanism, but it's cyclic AMP is a typical secondary messenger, which is cAMP. And so that is what eventually opens up the ion channel. And so that's much slower. So acetylcholine would be direct acting. Dopamine is actually indirect. So dopamine and serotonin are indirect. So you could write that down next to your acetylcholine is direct. Right? Dopamine and serotonin are indirect. Now, sometimes it actually depends on, we'll look at some other neurotransmitters besides. We have 50 different types of neurotransmitters. There probably are more than 50 that we only know about 50. And some of them, it actually depends upon the synapse, whether they're going to act directly or indirectly. So it's a little bit more complicated than just being direct or indirect. Sometimes it's the synapse that determines whether they're going to act one way or the other. Okay. So the other thing is the idea of excitatory versus inhibitory. So we have excitatory and inhibitory. So excitatory would be that it causes a depolarization due to the opening of sodium ion channels. Okay, so it becomes more positive. It moves towards zero. And remember that it has to hit that threshold before an action potential is generated. And then the inhibitory is hyperpolarization. And this is actually due to the opening of chloride ion channels. So chloride is more concentrated outside the neuron. So if you open chloride channels, then chloride ions will flow in and they will make the inside more negatively charged. And so that's how inhibitory neurotransmitters tend to function. Okay. So in your book, there is a table. And I'm just going to talk a little bit about this table. So when we look at acetylcholine, it is, as I mentioned before, excitatory with a direct action in most cases. However, in the autonomic nervous system, it can actually slow your heart down, and so it would be inhibitory. And when it is with the autonomic nervous system, like interacting with the heart, it actually has an indirect action. That's how you would read that table. So if we look at, one of the interesting things about acetylcholine is that the levels of acetylcholine get really low in Alzheimer's patients. And so one way to treat Alzheimer's disease is that they give them acetylcholine. So they give them that as a drug in order to hopefully it'll increase the acetylcholine in the brain. So if we look at some other neurotransmitters, we talked about dopamine. So here, note that dopamine can be excitatory or inhibitory, depending upon the receptor, that it depends upon that. And it has an indirect action. And this is your feel-good neurotransmitters. Dopamine, on the other hand, is actually deficient in Parkinson's disease. And so Parkinson's disease is due to a degeneration in the part of the brain that controls movement. And so people start to shake and they lose motor control. And so one way that they actually treat people with Parkinson's disease is to give them a drug that's called L-DOPA. And L-DOPA is a synthetic form of dopamine, and that tends to decrease the symptoms. It also increases in schizophrenia. So schizophrenia is associated with hallucinations and hearing voices. And so that could be due to the extra excitement of dopamine, the extra levels of dopamine in the brain. Okay. So if we move down our table, there is another inhibitory neurotransmitter in the brain that I mentioned previously, and that is called GABA. So GABA, so GABA. And it is actually the prime inhibitory neurotransmitter in the brain. And so it is one of those that has direct and indirect actions. And it opens up those chloride ion channels and actually causes hyperpolarization. So you can actually affect the way that the GABA works by taking alcohol. So alcohol actually augments the inhibitory effects of GABA. And so that's a way that alcohol suppresses the nervous system, as it actually allows GABA to work better at being inhibitory. Then we have a neurotransmitter called glutamate. And this is actually the most important excitatory neurotransmitter in our brain. So we talk about dopamine as being feel good, but glutamate is actually really important for learning and memory. And so your glutamate levels in your brain are going to determine how well you remember and learn certain things.
Is that a reason why people will maybe take like Adderall, or like does that help increase or decrease?
That's for hyperactivity, is that right?
Well, yeah, just how you were kind of saying like good for memory and stuff. Like I know some people, that me being included, I don't take tests very well.
Right.
So like people have recommended me going and taking Adderall or a beta blocker. And I was just curious if that's one of the reasons why, if like maybe some people don't produce that much of a neuron.
You know, I haven't looked at Adderall, so I don't know, but it might affect the levels of neurotransmitters in your brain. Yeah, I don't know that one specifically, but maybe I'll look that up and check that out, because that would be interesting to me.
I was just curious with all like how you're saying like dopamine, like
right? You
can kind of relate that to a street kind of related drug, I guess. And I was just curious if, I know there's people will say there's Adderall is like a form of cocaine. I didn't know.
Oh, interesting. So maybe
yeah. Maybe that's what it was.
Yeah.
So I just was curious if that also had something to do with sparking some of these other triggers or something along the lines of that.
Is there an outtake? Oh,
I'll have to check it out.
Okay, so then we have the endorphins, right? And they are generally inhibitory, and they act via an indirect action. And they are widespread in the brain, and they are natural opiates. And so they block pain perception. Okay. That can be a good thing. But again, if you're taking like heroin, which mimics the action of endorphins, your body is going to sense that you have too many endorphins. And too much endorphins means that you are not going to sense pain. And so that would be a bad thing, because we need to be able to sense pain in order to protect ourselves, right? So if you're not sensing pain, then your body is going to shut down the production of endorphins, and so that you're going to go through a wicked withdrawal if you go off your heroin, because your endorphin levels are going to be so low that you're going to be in a lot of just overall general pain in the body, right? And so that can be a really bad. So they talk about that the effects of endorphins are mimicked by morphine, heroin, and methadone. The reason why methadone is a better choice than heroin is that it is not used intravenously. They do not inject it, so it can be a liquid or a pill. And that means that the people that are taking it are a lot less likely to get hepatitis or HIV from using intravenous drugs. Okay, so let's look at one more table here. Okay, so this one is actually something that has been added to your textbook just recently, because it's just been recently kind of discovered. And this is called the Endocannabinoids. What do you think that is like? Why are they calling it a cannabinoid? Cannabis, right? So this is marijuana, right? So actually, we have, just like we have endogenous morphine, we have endogenous marijuana in our body, right? And it is inhibitory, and it's throughout our central nervous system, and it is also involved in memory, appetite control, interestingly. I'm not sure how that works. But nausea, vomiting, neuronal development. And so this is really of interest, because people are thinking that they can use medical marijuana to treat maybe some of the nausea, vomiting effects, or sickness from people who have cancer and are doing chemotherapy. And then also some of the pain, because it's inhibitory. So maybe chronic pain can be better treated with marijuana than it can be treated with morphine or other opiates.
Yeah, my friend who has had leukemia, they recommended him do cannabis because all the medicine that they were giving him, he was getting addicted to.
Right. Yeah.
It's making it easier.
And so you can actually get the tinctures or the oils that do not have any of the THC, but also just have the CBD. But notice that this one these are actually the same receptors of those endocannabinoids that are activated by THC. And so it's kind of interesting, you know, we can get, I actually know somebody who, one of my relatives, on my husband's side, moved here from Alabama because her daughter had seizures, and she had seizures like 10 a day. And she moved up here so that they could give her CBD oil, and she has had in the last 2 years, she's had like 5 seizures total, right? So the use of CBD oil has dramatically affected their life because now she can grow and develop a little bit better than when she was constantly in a state of seizure. Okay, any questions about those? Okay.
I have a question.
Yeah.
The ones that we went over, are those the only ones that we need to know?
Yes. So you need to know the endorphins, the endocannabinoids. Oh, we didn't talk about norepinephrine, so we need to know that one too. But we're going to talk a lot more about these when we get to the autonomic nervous system. So norepinephrine is actually influenced by methamphetamines. So it is also a feel-good transmitter, and it gives you energy. But also the withdrawals from that can be very bad too, where you go to sleep and you can't wake up because you have such a low levels of norepinephrine. Okay, yeah.
Does that work the same way as like cocaine?
Actually, methamphetamines have a different effect, which is actually worse than cocaine, in that it causes excessive release of dopamine. So it causes dopamine to just be released excessively, and it can actually lead to the death of the neuron. And so you can actually get nerve cell death in the brain, which causes people, even after they come off of the methamphetamines, even after they come off of the withdrawals, they'll have shakes, and they won't be able to stop shaking. And that's because they've actually done damage to the part of the brain that is the same part that affects Parkinson's disease. And so, I should mention that ecstasy is really bad too, because it also leads to the death of neurons, specifically serotonin releasing neurons. It causes excessive release of serotonin, and then it causes death of those, and so it causes essentially brain damage when you use it. Okay. So let's talk about some terminology that we're going to use as we talk about the nervous system, the central and the peripheral nervous system, because we're moving on to chapter 12 today. And when we talk about the nervous system, we can talk about what is called the white matter and the gray matter. And you saw in lab that they do have distinctive colors, right? They're actually maybe not gray, it's actually like maybe tan colored, I think. You know, white is definitely white. But what makes the white nervous tissue white? What is it about the nervous tissue, like say, for example, in part of the nerve cord, that makes it white and not colored? Not collagen, that's a good guess though. No. It is the myelin. So these are where we have myelinated axons. So we know where we see white matter in the brain and in the spinal cord, that's where we have these myelinated axons that are running in groups together, in bundles together through the nervous system. So the gray matter would be the cell bodies and the dendrites. That would be your gray matter. We can also talk about tracts versus nerves. So tracts are bundles of axons in the central nervous system, whereas the nerves are bundles of axons in the peripheral nervous system. So we'll say that they're both bundles of axons. Tracts are in the central nervous system, the brain and the spinal cord. Nerves are in the peripheral nervous system. And it's important to realize that nerves are actually organs because they don't just consist of nervous tissue. There's also connective tissue and other types of tissue that surround it and bundle it. And so nerves are actually considered to be organs. As are individual muscles, like your biceps muscle is also an organ, and even your blood vessels are considered organs because they're just not made of one single type of tissue. Okay. We can also talk about nuclei and I'll put nuclei versus ganglia. So nuclei, you want to get away from the idea that it is a nucleus in a cell because these are just aggregations of cell bodies. So in the brain, we have nuclei like the basal nuclei. So this is an example, the basal nuclei. And this is gray matter in the middle of white matter. So it kind of looks like a nut inside the white matter. And so that's why they call it a nuclei. It's a nut. So it is embedded in white tissue. So it's just an aggregation of nerve cell bodies. Okay. We also have ganglia. So this is in the central nervous system, CNS. Okay. So we have ganglia that are aggregations of nerve cell bodies in the PNS. Okay, so ganglia are in the PNS. So we have these ganglia that are right next to the spinal cord, and then they send the axons out to, say, for example, the blood vessels, to the heart, to the sweat glands, right? And they're aggregations of nerve cell bodies that exist out of the central nervous system, outside. Okay. So today we're going to start talking about the brain. So when we look at the central nervous system, it is actually derived embryonically from ectoderm. So it's derived from embryonic ectoderm. When we look at the muscles, that's actually derived from mesoderm embryonically, and as is your skeleton, is also mesoderm. So ectoderm is outside. And so when we start off as an embryo that has layers. We have the outer ectoderm, the middle mesoderm, and the inside endoderm. And so this ectoderm actually becomes our nervous system. So this ectoderm rolls into a neural groove. Actually, let's talk about the neural plate first. So you have a neural plate. It develops into the neural groove. And then it develops into what is called a neural tube. And you might have heard of the neural tube. because there are certain birth defects that are called neural tube defects, birth defects. And so we'll talk about spina bifida is one of those where the neural tube does not develop properly. And so there's part of the spinal cord that extends outside of the vertebral column and is not protected by the vertebrae. So if we look at a diagram of this progression of development, right? So this is my neural plate. This is my ectoderm, right? It develops into a neural groove, and then we get what is called the neural tube. Okay, so this occurs very early on during embryonic development. The important thing here is that you'll notice that the neural tube is hollow, and it's filled with cerebrospinal fluid. Okay. So the neural tube, hollow, and it is filled with cerebrospinal fluid. And you can abbreviate that as CSF. Cerebrospinal fluid. Okay, so when our neural tube develops, we have an anterior or head end, and then we have the tail end. So the anterior end is going to then develop into the brain, and the posterior or the tail end is going to develop into the spinal cord. And so depending upon where the cells are located, then you get the specialization of the different parts of the brain. And so when we look at the development of the brain, okay, we want to talk about what is called the prosencephalon. So encephalon just means brain. So when you talk about encephalitis, that's infection of the brain, right? Microbes, viruses, bacteria. So prosencephalon is the very first part of the brain, okay? So you need to know that there's two parts of this first part of the brain. So I'm going to branch it into two, okay? The one that's at the very end is called the telencephalon. Okay. This is going to become our cerebral hemispheres. So this is the biggest part of our brain. 80% of our brain is in the cerebral hemispheres. So when we look at our cerebral hemisphere, it's very big, and it actually has, during embryonic development, folded back in on itself. So there is another part of the prosencephalon, which is called the diencephalon. And this includes the thalamus. Oops, I spelled that wrong. Thalamus, M-U-S. The hypothalamus, which is underneath the thalamus. And what is called the epithalamus, which is a pawn up on top of the thalamus. Okay, so that's my prosencephalon. So in your book, they show the development of this. So this is the neural tube. Notice that the anterior end of the neural tube gets big, and then it develops into the forebrain, which is the prosencephalon, develops into the telencephalon and the diencephalon. So we also have a middle brain and a hind brain. And notice that these have been broken down into subcategories, but you do not need to know those because they're smaller. So you need to know the telencephalon and the diencephalon. The midbrain is just the mesencephalon, and the hindbrain would be the rhombencephalon. So let's write those down. So I'm going to put, this is my forebrain. We have the mesencephalon. Bencephalon. That's an O. This is my midbrain. And then we have what is called the rhombencephalon. And this is my hindbrain. So the hindbrain includes the pons, which we'll talk about, the medulla oblongata, and it also includes the cerebellum. Easy to confuse cerebellum with cerebral. The cerebellum is kind of a mini brain that sits back here, and it is part of the hindbrain, not part of the forebrain. And then notice that the pons and the medulla oblongata would be in direct connection with the spinal cord. So from there, from the hindbrain, we connect to the spinal cord. Okay, so this would be the spinal cord right here. So notice that these are fluid-filled chambers. And so the fluid-filled chambers are actually called ventricles. Okay, so those ventricles are filled with fluid and they help to bathe and provide nutrients to the cells. They also kind of add buoyancy to the brain. And kind of, we'll talk about how it also protects the brain a little bit when we get to the meninges, the outer covering of the brain. Okay. So if we look at these ventricles, these are, oh, wait, before we talk about the ventricles, I wanted to show you this one. Okay. So the reason why our forebrain is so developed and also encompasses actually the midbrain is the way that our skull limits the growth of the cerebral hemispheres. So when the skull develops, you'll notice that's my telencephalon in kind of tan. It actually develops and it starts to fold back over the rest of the brain. And so that's why the midbrain in us, not in all animals, is in the middle of the cerebral hemispheres. And then something else really interesting happens in our brain that doesn't happen in all animals is that our cerebral hemisphere starts to be convoluted so that it has ridges and grooves on it. So notice that my cerebral hemisphere covers the diencephalon. So this would be like my thalamus, my hypothalamus. This is actually my pituitary gland, so it's like in the middle of the brain there. It actually comes off. And then this is my midbrain, and this is the pons and the medulla oblongata. So this is the cerebellum. So it is separate from the cerebrum. And so we're going to talk about those ridges and grooves, but that kind of gives us an idea of how much of brain area is associated with conscious thought. And so when we look at some animals, like birds, right? They do not have ridges and grooves. So the idea is, well, maybe they do not have as much surface area for conscious thought. But recently, they have discovered something very interesting about bird brains. They couldn't figure out how crows and ravens and corvids could be so intelligent. And so they re-examined the nervous tissue of birds, and they found that their nervous tissue is more highly concentrated than ours. And so if you looked at just a given area of unit of area, they would actually have more neurons in a given area than we have in a given area of our brain. And so they think now that crows probably have the same mental capacity as cats and dogs. Because of the increased density of neurons in their brain. Okay. So also, if you look at dolphins actually have more surface area. And so if you look at their outer cerebral hemispheres, they're actually more greatly convoluted. And they have about the same amount of nervous tissue to body size as chimpanzees, but we still have a little bit more nervous tissue in terms of our nervous tissue to our body size. So they have just more, their cerebrum is more convoluted than ours. They also have these really interesting characteristics where their visual organs are associated with their hearing organs, so that when they use echolocation, they can actually turn that signal into an image. So when they're echolocating by sending out sounds and then receiving the sound waves back, they actually see images when they do that. Okay, so now let's talk about the ventricles. Okay. So we have the ventricles. We have the two lateral ventricles. And these extend into the two cerebral hemispheres. Okay, so if I go back to my diagram. Oh, actually, I think I have a picture here. Oh, I don't, sorry. Didn't organize that correctly. Okay, so if we look at the lateral ventricle is going to be in the two cerebral hemispheres. I know I have a picture of this somewhere. And then we have a third ventricle, so that's 1 and 2, since there's 2 of them. We have a third ventricle, which is in the diencephalon. And then we have this little tiny cerebral aqueduct that goes down through the midbrain. And then the fourth ventricle is associated with the hindbrain, the brain stem. So 2 lateral, 1/3 ventricle, the cerebral aqueduct, and then the 4th ventricle. So that's a diencephalon, that's a C cephalon. And then the cerebral aqueduct, which would be the midbrain. And then the 4th ventricle, which is in the hindbrain area. And these ventricles are continuous with the central canal of the spinal cord. Okay. So if we look at the cerebrum, we have 2 hemispheres. And these have what is referred to as lateralization, which means that we have the right and the left. So most of the sensory information that is coming from my left side of my body goes to my right hemisphere. So it crosses over at some point and it is integrated on my right side. So the left body is perceived on the right side of the brain. And then the right side is perceived on the left side. And then also they believe by looking at what are called PET scans, where they can actually give a person an activity, and then they can see where in the brain there is the most electrical activity in the brain. They believe that the right side of the brain is actually more associated with creativity, right? Also, language. So when you're writing something, maybe writing an essay or a poem, it's the right side of the hemisphere that tends to light up biggest in electrical activity. And then the left side tends to be more logic, analysis, and math, I would say. So when you're solving a math problem, right, and trying to logic something out, then the left side of the hemisphere would light up for you. These two cerebral hemispheres are connected by a group of white tissue, which is tracks, right? So it's connected by the corpus callosum. And this is white matter. This makes up, the cerebral hemispheres make up about 80% of the human brain. However, only the outer part of the cerebrum is associated with conscious thought. And so this is the cerebral cortex. So this is the thin outermost layer of the cerebrum. And it's only believed to be about 4 millimeters, which is like half a centimeter in thickness. And this is gray matter. So therefore, it is nerve cell bodies. So weirdly, our nerve cell bodies are on the outer part of our cerebrum, and then their axons extend down into the middle part of the cerebrum, which is considered white matter. There are some nuclei, however, in the brain where we have aggregations of those nerve cell bodies outside of the cerebral cortex. Okay, so this is what is responsible for conscious thought. This also has a large surface area. And the reason why it has this large surface area is because of the ridges, which are called gyri. The gyri, or a singular would be gyrus. So a single ridge would be a gyrus. The ridges in general are called the gyri. Okay. And then we have grooves, which are called the sulci. That's just one eye, sulci. Or a singular would be the sulcus. Sometimes we have big grooves, and so large grooves are called fissures. So, for example, there is a large longitudinal fissure that runs between the two hemispheres. And so there have actually been people that have had their corpus callosum cut and the connection between their two hemispheres completely severed, and they are able to survive. That is due to or done to people that tend to have really bad epileptic seizures that are life-threatening. Sometimes they'll actually cut the connection, so they'll go down through that large groove and cut the connection between the two cerebral hemispheres. So if we look at a picture of this, okay, that would be the longitudinal fissure. All of these others, little fissures, would be sulci. And then the ridges are the gyri. So you'll notice that the cerebrum is actually broken into different lobes. And these lobes correspond to the name of the cranial bones. And so in the cerebrum, we have 5 lobes. Okay, so we have the frontal lobe, which is near the frontal bone. There's actually 2 of these, right? They are on each side, right? So there's 2 frontal lobes, the left and the right. Then we have the parietal. It's the same for all of these. There's 2 on each side, or 1 on each side. So that would be near the parietal bone. Then we have the temporal bone, or the temporal lobe. And then we have the occipital lobe. And those are all the lobes that you can see on the outside. So there is another lobe. There's a 5th lobe, but it is hidden on the inside of the brain, and it is called the insula lobe. So it is on inside. So it's kind of near the temporal lobe.
Is there 2?
Yeah, there's 2 on each. So there's frontal lobes left and right, parietal lobes left and right, temporal lobes, and then occipital lobes back here. So I think there might be a picture of the insula lobe here. Yes. And so if you look at these lobes, this would be the frontal right here, the temporal, the parietal, the temporal, the occipital. And then if you look inside, you would be able to see in here, there's actually some folding ridges and grooves that have actually become incorporated into the inside of the brain. And that would be the insula lobe. Okay, so we're going to talk on Wednesday about the function, just briefly, of each of these areas of the brain. And so this is where we can consciously perceive different stimuli. So like vision goes to the occipital lobe. For hearing, auditory would be near the temporal lobe. Okay? So the lobes are associated with perception of different stimuli.